So as it breaks other genes, we can tell which gene was doing what. So that was the hypothesis when we started this project. So very easy to follow this because you are growing this plant. If you started seeing any plant with purple color flower, that means transposable element jumped out of the flower color gene. So that is the only plant of interest. You don't worry about all the plants which are variegated flowers. You have to just follow the plants where it jumped out. So I will show you that in animation, that this is transposable element, and this is the flower color gene. If this jumps into this flower color gene, it breaks the gene, now the coloration will disappear. It becomes white color. But as it is not stable, it is going to jump out of here too. So in this case, it jumps out from there. When it is out of this flower color gene, this color should come back. Because it is out of that, this gene is intact. When that happens, when you see purple color flower, that means in that plant, transposable element jumped somewhere else. So you have to find where. In this example, it jumped into fertility gene. So it broke the fertility gene. Now this plant, which was fertile here, not fertile anymore. It is sterile. So that is the main mechanism we were trying to follow. That was the approach we were trying to use. So you start with plants with variegated flowers and grow next generation. Most of the plants are going to be variegated pattern, except for few, 5, 7 plants out of 100, where transposable element jumps out, those plants will turn purple. So we don't worry about these white flower plants, we worry about this purple. So if it jumped out of this purple color gene, where is this going? So you look at the progeny of these plants. Is there any problem in the progeny of these purple color flower plants? If there is any problem, that means that problem is caused by a transposable element. So that's how you follow that gene wherever it is going. So when we screened large number of plants like that, we found several different types of characteristics where the plants were lacking something. The first one was necrotic root mutants. The plants, they started making roots which were yellowish in color, looked like they are diseased roots. Then the second type was chlorophyll deficient mutants. So chlorophyll is the pigment, green color pigment, which is present in the plants. So if something goes wrong with that gene, then plants will appear yellow in color. So these were very yellow plants. And some of them, they were lethal. Some of them, they are viable. They can still live because there are other pigments present. And the third group, fertility mutants. There were several different types of fertility mutants, and this is the part we are most interested in. So when we looked at fertility mutants are 4 big groups. So one, there are partial steriles, partial female sterile, partial male sterile. And then there are complete steriles. In this, there are male steriles, but female is okay, female is fertile. In this, they are male sterile and female sterile. So these are the mutants we named, which we identified. So these in color here, these are different types of mutants we have identified in each of the groups. Now, big question. Why are we interested in fertility? Fertility is a very important process during sexual reproduction in plants. More importantly, fertility can be instrumental in developing hybrid soybeans. And I'm going to emphasize on that a little later, that how we can develop hybrid soybean using fertility. Hybrid plants are better than pure lines. So there are many different types of hybrids in vegetables. Corn hybrids are very famous. There are so many hybrids in rice. So hybrids, they produce a lot better than the pure lines. Then also, in addition to that, will learn the network of all the genes which are involved in fertility in sexual reproduction, which is going to be big addition to reproductive biology of soybean and also to other plants. So hybrid soybean. What is the problem with hybrid soybean in soybean? You look at the flower of soybean, male and female reproductive structures are present in the same flower. It is self-fertilized. If you do not do anything, the male reproductive structure will produce pollen grains. Female reproductive structure is right there. So pollen grains will fall on female reproductive structure. Still flower is closed inside the flower. So it will self-fertilize. It will produce a cell which will develop into an embryo. So it will make seed. So it is self-seed. When you are talking about hybrid, you are talking about one plant cross to other plant. That's how you make hybrid. You take pollen from one plant, put on reproductive structure of the female plant, and then the seed which will be produced, that is contribution from two different plants. That is hybrid. But soybean, to produce hybrid, what you will have to do is, you'll have to, this is the flower bud, very, very tiny, this much. You have to open up this flower bud, and inside that, these are male reproductive structures, and this is female reproductive structure. You'll have to cut out the male reproductive structures with scissors. So if you cut these out, you have only female reproductive structure remain here. So here you can see it better. Then you take another flower from different plant and take pollen from there and put it here. And then when it will form seed, that is going to be hybrid seed. This is my collaborator, Dr. Reed Palmer, sitting in the field, trying to remove the male reproductive structures from the flowers, from the female flowers. Guess how many can you do in a day? So if you want to produce hybrid soybean, farmers are going to ask for the seed. Give us that many million tons of seed so we can grow it. So you can ask me a few pounds, but not million tons. So that's the big problem with soybean. You cannot produce hybrid seeds. So now how fertility can be instrumental in that? If you have plants or which will produce flowers which are male sterile, they don't have viable male reproductive organs. So here you look at male reproductive structure, it is kind of shriveled. It does not produce viable pollen grain. Female structure is fine in this case. So this is male sterile only. So then you have other plant where male and female both are fine. So you don't have to remove the male reproductive structure from here. You can plant both of these right next to each other. So one line of male sterile, one line of male fertile right next to that. And then insects can help you out. They will take pollen from here, transfer pollen to this plant. You don't do anything. You harvest seed from this, not from this. Because this is going to be self, you harvest seed from this, all the seeds set on this plant is going to be hybrid seed. So that's how you can utilize fertility to develop soybean hybrid. So now I'm going to tell you a story about one of the mutants which we characterized in a lot more detail. And in the middle, I'll tell you a little bit about others also. So we identified, in the beginning I told you we had 4 different types of mutants. You have partial steriles, then you have male steriles, but female fertile, then male sterile, female sterile. Out of that, there was this mutant which was male sterile and female sterile. So we called it as ST8 mutant, ST for sterility, sterility 8. So we already had up to 7, so that is number 8 in that class. Look at the fertile plant. They were sown at the same time. So this is fertile plant. This is sterile plant. Sterile plant does not produce any seed. If you look carefully, where the flowers are present, in that part, it will produce bud-like structures, without any seed. So we wanted to characterize that. For characterization, first step is you want to know which gene is making it sterile. And you want to know location of that gene on soybean chromosomes, where in the DNA this particular gene is present. So we call it as mapping. So you want to map the gene in the soybean chromosome. And we use one approach called as bulk segregant analysis. I'm not going to go into too much technicality. I'm not going to explain what this approach, how it works. But I'll tell you overall that you take mutant plant, you cross with fertile plant, and then you produce next generation, which is, we call it as first generation, F1 generation. And then you let it self-fertilize. You don't do anything, produce next generation, which is called as F2 generation. So in second generation, you will have some plants which are going to be fertile, some plants are going to be sterile. So what you do is, you take some pure fertile plants and pool them together. You take some pure sterile plants and pool them together. So we isolate DNA out of those plants and pool them together in one tube. So we call it as fertile bulk, and this as sterile bulk. So the basis behind this approach is that you look at the markers from throughout the soybean genome. Markers are something which we use to map gene in relation to. So some DNA-based markers we use to map gene on the chromosome. So when you use these markers, every single region in the genome of fertile and sterile should be same, except for the region where you have fertility gene, it's going to be different in fertile. Where you have sterility gene, it's going to be different in that bulk. So everywhere else, it's going to be same. So when we run these markers, so we have about 800 DNA-based markers in the lab. So this is first marker, run on fertile bulk and sterile bulk. When you see these two bands right next to each other at same level, that means they are not different. So they are same here, they are same here. When you see it as same, means this is not interesting. Gene is not present close to this marker. When you start seeing something like this, you have fertile bulk, sterile bulk. This band is a lot lower than this band. So that is different. And then you have next one, this band is a lot lower than this band. So these two markers are really markers of interest for us. So I put the names of these markers up there. So these are the names of markers. So we know that our gene is present close to these two markers on soybean chromosome. And we already know the location of these markers, where these markers are on soybean chromosome. Then we go to that part on the chromosome, look at all other markers present close to these markers, and run those markers on entire F2 second generation population. So we had about 96 plants in each population, so in F2 population. So we run on all 96 plants to see the pattern. Plant 1 is giving you this kind of pattern, plant 2 is giving you this kind of pattern. So here you look at parent 1, lower band, parent 2, higher band, and these are all F2 plants. In F2 plants, either you are going to see this lower band, or you are going to see this higher band. Sometimes you will see both of them. So that is the pattern you are going to get. So these are plant number 1, 2, 3, 4 going like that, and you record that pattern, that what kind of pattern you get on each of the plants. And then you try to match that pattern with the character, whether it is fertile or sterile. Closely it matches with the pattern, closer it is to the gene. Again, I'm not going to go into too much detail of how we do that, but we get a map of the chromosome, which will show you these markers. And this is our gene of interest, that this gene maps close to this particular marker on the chromosome. So this, we mapped this, but this is pretty loosely made map where you see markers pretty far away. So look at this marker pretty far away from the gene. So here, this is pretty far away too. But we had to map many mutants. So as I showed you earlier, we had 12 different mutants identified of different types. So then undergraduates come handy. So with the help of over a dozen undergraduates, we were able to map all of them. So these are different maps for different genes: male sterile, partial, female sterile, male sterile, male sterile, female sterile. So these are few of those, and remaining are here. So we were able to map all the genes we identified to soybean chromosomes. And we were able to publish this work in refereed journals, scientific journals, where undergraduates, they were first, second authors, and pretty good contribution from the undergraduates in this project. So now, you map those. What is the next step? Next step is going closer. You have a map which is very low resolution map. So like we have maps of cities and stuff, you want to go closer. So at street level, where this particular location is. So then we developed lots of markers close to the region where the gene is present. So these are all new markers on the same map, the map which I showed you before. So all new markers present. Now you look at this gene, 2 markers present right next to that, 0.4, 1.2. So that is distance, relative distance shown there. It gives us complete idea that our gene is present between 428 and 430, these 2 markers right next to the gene on both sides. So our gene is present in this part. From there, when we were so close, we had to jump onto the sequence. So I was part of the sequencing effort. Soybean genome was sequenced in 2010, and there were like 30 different labs involved in sequencing soybean genome, and our lab was part of that sequencing effort. So from there, you go to the sequence information. You look at these 2 markers, 428 and 430. On the sequence, every single base of the DNA you can count, and it is between this and this physically on the sequence map. I'm going to show you even zoomed part of that. So that is these 2 markers, 428, 430, gene is in there. So you go to the sequence map here and you zoom that out, see what is present in this part. Look at all the DNA sequence in that part. There are 5